Yeah, so that's the title of the talk, Morphogenetic Decoding of Positional Information, and I'll tell you what that means. Uh, so the talk actually, since to prove that I actually attended talks last week, this talk is going to be very similar, close to the talk given by Massimo and Andrelio. Okay, so there'll be more on uh, inference, statistical inference, and information theory as applied to developmental biology. Yeah. So okay. So yeah. So the the problem that organisms need to face during development is that development, when it starts with one single cell, has to reproducibly and reliably form an organism with its different organs. Okay, and it has to do it at specific uh, sort of spatial locations. You can't have a hand somewhere on your head and so on. So it has to be very reliable, and we'll see how reliable it needs to be. Okay. Now, this reliability has to be attained in the presence of a huge amount of noise within the system and also environmental noise. Okay, so that's the challenge that every developing organism needs to face. Um, in order to do this, what the developing organism does is that its cells need to collectively talk to each other, and it's through an interplay of information, mechanics, and geometry that these reliable uh, the reliable development occurs. Okay. One of the key points in development is that a cell needs to be able to figure out its position from some origin. Okay. This cell, let's say one cell somewhere far away from the putative head, has to know that it has to be the thorax, eventually the thorax. Okay. So it needs to know and infer its position. And that's what I'm going to talk about today. How do cells in a tissue or in a growing embryo infer their position accurately, despite the fact that there's a huge amount of noise? Yeah, so that's the, okay. And to do this, um, I'll, so we've been working on these ideas for some time in different contexts. And what I'm going to talk to you about today is this decoding of molecular information. Okay, I'll explain what this means. These are the two papers that have been published in these in this topic, and the primary author of this paper is my student Krishnan Iyer, who's a theorist and who's collaborated with Jitu Mayer's student Chaitra Prabhakar to and it's a sort of nice theory and experimental collaboration. That's what I'll show you. And this is the cartoon of what I just said. Here is a tissue made up of a collection of cells which are adhered to each other. This group of cells in this tissue declares the origin. Okay, that's the origin. And you know it's the origin because it's produced some protein, which is then secreted into the extracellular milieu, which then diffuses and is presented to each of the cells in the tissue. Okay, this is a very noisy signal, and cells in this tissue need to infer their position from this noisy signal. Okay, so that's the task. And what this paper is about what are the strategies that cells need to have in order to attain this task. Okay. The other thing which I won't be able to, most likely I won't be able to discuss, but is in the same kind of philosophy that it uses, is about, instead of decoding molecular information, you have to encode molecular information. And this is in the context of the cell, where all cells in the organism are coated with some complex molecules on the cell surface, which are synthesized by the cell itself. And the array of molecules that reside on the cell surface is a declaration of cell type identity. Okay, so it's a molecular code that every cell in the organism presents to the organism. And both these these topics, namely decoding of molecular information and encoding of molecular information, involve high dimensional optimization. It involves a dimensional reduction of stiff parameters. I'll come to this, what this means later. And it involves the interplay between information processing and systems biology that I'll talk to. Okay, so I just wanted to show you that these two topics, though they might appear as very distinct, are actually belong to the same kind of philosophical questions that we've been asking. Okay, so now coming back to the first topic. Okay, so this is a of course famous quote by Louis Wolpert, who described the inference of position of cells in a tissue in the following way. Okay, here you have a tissue group of cells. They look identical, but these set of cells are now produce a certain molecule called morphogen, and this morphogen is secreted outside in the extracellular space here. These are the receiving cells. They receive different amounts of this morphogen. And depending on the amount of morphogen that is received by these cells, depending on how far away they are from the source, they attain different fates, which eventually, you see, these cells have little apical hairs on the top. This has a little big sprout coming out. So the cell fate is declared by how these cells have inferred their position from this source. Okay. Yeah. So that's what I am suggesting here. And now the way these cells know their position away from the source is through this sort of morphogen that comes out. Okay. This morphogen that comes out has a certain profile, and the profile I'll show you later has, let's say, an exponentially decaying profile. So it's from this kind of exponentially decaying profile that these cells need to infer their position. Yeah. I'll tell you what exactly. Yeah, this is by diffusion, but I'll get to the exact.
No, I'm just wondering, it's just a continuous profile, whatever dynamics. But then there seems to be some very specific site something is coming up, which is not like, like when you have this, something is happening there, or some 5 things are of purple color. This seems to be very specific. This thing, right? It's not 5 as opposed to 7 or something, right?
Correct.
So how can like diffusive, which is like a coarse grain thing, can actually make so specific thing?
Exactly, that's the point. Yeah, exactly. So I'll come. That's exactly the point that I'll come to. No, what I do? Yeah. Okay. Yeah. Okay. So this is experiments that have been done to measure exactly what you're asking. How precise does this inference of position need to be? Okay. And so this is in the context of a Drosophila embryo, where it happens in every little organism that you can think of. Okay. So these are the dimensions. The length of the embryo, the Drosophila embryo, is 0.5 millimeter. Each the cell, and it contains around 100 cells. Okay, along that axis. So the cell size is about 1% of the embryo length. Okay, so this is the morphogen that's secreting, that little red dots that I showed you. Okay, this is a morphogen called bicoid. It secretes it from some producing cells here, and it has this exponential decay. Okay, now by means of this exponential decay, by reading each cell reads this exponential decay, okay, and says, okay, I belong to the front of the cell, the apical part of the cell, or I belong to the back of the cell, the posterior part of the cell. Okay, and that boundary, okay, that declaration happens through the expression of hunchback, which is some molecule. Okay, this boundary at which you declare that these are front and these are back is very accurate, and it's accurate up to 1 cell width. Okay, that's the accuracy it has. Despite the fact that bicoid is diffusing, it is very noisy, etc., etc. This, this step happens with an accuracy of 1 cell width.
It is exponential. There is fluctuation in the system.
Exactly. Those are exactly the questions I'm going to address. Yeah. Okay, this is experimental data, and I'll show you other experimental data as we go on. Okay. Now, this is in another context. I just want to show you that this happens in the early embryo with bicoid. It happens later on downstream with some other genes called even skipped, which goes to define the segment boundaries in the Drosophila embryo. Again, these boundaries, which are represented by peaks of this particular gene, okay, are accurate up to 1 cell width, and that's the experiments in this paper, which shows that the variance divided by the length of the embryo is around 0.01, which is around 1%. I mean, the error, that is 1% of the length, okay, 1 cell width. That's the accuracy. So there seems to be enough evidence in the embryo and enough evidence in the wing imagine in the larva of the Drosophila to say that all these boundaries, the boundary like I showed you earlier. Oops. No, there. Yeah. All these boundaries that I showed you here, they are so accurate, and they are accurate up to 1 cell width. Okay, in the embryo, in the larva, and everything. And the question is, how exactly does it happen? So this is the surprise, right? Now I do. Okay. So now I'm going to pose the question in a mathematical way. Okay. Now the question is the following. Here again is the tissue. It is presented with this morphogen gradient. As you said, the morphogen is very noisy, right? And so every cell is going to read out its every cell at a position x away from the source is going to read out its ligand distribution. So I'm given a P of L given x. Okay, what is the probability distribution of the ligand concentration, local ligand concentration given the position from the origin? The cell is going to receive that, okay, by molecules that sit on the cell surface, and is going to sort of process the information. And the processing could be complicated. It could be happening at the cell surface, in the interior. We'll allow all these possibilities to happen. In fact, they do happen. Okay, and as a result of reading that noisy information, what happens is that a gene is transcribed. There's some transcription, and the gene is transcribed, and that is the output. So this is the input, a noisy input, and would be a noisy output. Yeah, and it's from this output that the cell has to infer its position. So the flow chart is this: at every position x, the cell reads its ligand P of L given x. It then processes that information through a channel, okay, which involves the cellular machinery, etc., and then gives rise to an output, okay, a signaling output, which is some transcription. From the signaling output, it has to infer its position. Yeah, and the question is, how accurate is that position be? How accurate is this inferred position from the real true position of the cell? Okay, yeah. That's a good question. In reality, it is a function of time. But in what we're going to do today, we'll take it to be, that time scale is very fast. So you get a steady state profile, which is established very quickly. Yeah? I'll show you the equations that are usually used to describe L. Yeah? Okay, and now it has to make this inference. It has to make this inference of position in the presence of noise, and the noise resides both in the input and in the fact that there's actually a chemical machinery going on, which is also noisy. Okay, the internal mechanism is noisy. So there's an external noise, and there's an internal noise. Okay, now, right. So here is the morphogen decoding problem that we want to establish by using an information theory framework. And here it is. We want to say this is the objective that the cell wants to do. Given an input, namely the morphogen ligand distribution, P of L given X, what are the requirements on the information processing, namely the reading, the information processing, etc., all these steps, such that the inference of the cell position, namely X star, through its transcription readout, is robust to both extrinsic and intrinsic noise. Okay, so now this is an attempt to formalize what we just discussed. Okay. No, how do I do this? Yeah. Okay. So here, so this recalls some of the stuff that Massimo talked about. Yeah? Okay. So you've gone through this. So what we are going to do is that since the noise and the channel, both the channel and the noise is noisy, and you don't know the details of what is really happening in the system, we are advised to take a statistical or probability distribution point of view. And we, in fact, will have to invoke Bayes' theorem, okay, in the following way. Remember, what you're getting is this morphogen, namely P of L given X. Then the cell machinery has to interpret what that morphogen is. Right? It has to convert it to an output, its transcription readout. Okay, so therefore it gets an input of this, and what it has to convert it into is a cellular output, namely P of the output, P of theta given X. So P of theta given X is a function of both the cellular machinery and how it reads the input, the noisy input. Okay, P of X here is the prior distribution of the cellular position, which you can take it to be uniform.
Madan,
Yeah. Yes.
where does the cell store this information, P theta given X or P X given theta?
Right. So P of theta is generally a transcription readout. The signal comes from the outside, eventually goes into the nucleus, and it turns on some genes, and that's the output. Yeah.
Not as if a single morphogen molecule is coming and hitting the cell. Then the errors would be just huge. So this is the probabilities are based on certain number of cells hitting, and each one of them is going through this process.
Absolutely.
And based on that, some inference is being made about the distance from origin.
That's right.
So there'll be a statistical average over that many morphogen molecules, and that is where the probabilities. Yes. Okay.
Exactly. Yeah. Okay. So fine. So this is the invoking Bayes theorem to calculate this P of X given theta. That is what the cell has been doing this using its machinery, but I want to now use the output to infer what the position is. Therefore, I want to now use the output to say what is my estimate of my position away from the origin. Okay, and that's what this is. Yeah, and then I'm going to use again. You must have heard this in Massimo's lecture. In order to extract what the inferred position is, I'll use an argmax of this distribution. Okay, so for instance, I'm going to record this distribution at various instances, and I'll take whatever is the inferred position. I'm going to record that and establish my curve P of X given theta, and I'm going to declare as X star the position inference. What is the value of X at the maximum? Okay, so that's what argmax does. So maximum a posteriori estimate. Yeah. So it turns out that if you assume that P of X is for cases where you have a prior which is uniform, then this can be provably shown to be most optimal. Okay, it's much more optimal than the mean, etc., etc. Fine. So this is going to be the task of every cell. Every cell is going to construct this X star by taking the input, using the channel to compute the output, use Bayes' theorem, or infer the position by this argmax. Is it?
You're basically going to apply the noisy channel coding theorem in the biological context.
Yeah.
But is there a mechanism for error correction if the cell realizes, oh, I'm off by 2 cells, or 3 cells?
Right. That's something that we are thinking. Let's hold that thought and we can come to this later. Yeah. So this is our first attempt at trying to, but many of these questions that you're asking are relevant. Okay. So is that clear? Yeah.
Find this X star.
Yeah.
The cell should know the distribution across, right? Not only cell should know what is happening at X star delta X, X star delta.
So a cell. So this is purely a localist declaration.
No, X star is.
X star is the value of X that the cell is going to record by having constructed this P of X theta.
This is only
local. Yeah. I'll maybe when I come to the next state, this thing I'll tell you. Okay. So this is what every cell is doing.
Because I thought that X is X goes from 0 to L, right?
Yeah, exactly. Huh? Yeah.
But then if I'm sitting at some position X, one cell is some position X, right?
Yeah. I don't know what X is.
I know what X is, but how am I going to?
No, I don't know what the cell doesn't know what X is.
Cell doesn't know where it is sitting.
Where it is sitting, okay, right? But it can read the local concentration of the ligand, and that is to estimate its position from reading that noisy concentration of ligand. That is the task.
No, this argmax, how are the cell going to compute just based on local?
So locally is going to compute, this is all statistics, right? So it's going to collect at every instance values of L, it'll compute theta, you'll get a distribution of theta, you'll get a distribution of L, it'll compute P of X given theta, and then plot this. Obviously the cell doesn't do it, but it'll plot, and then it'll estimate the value from this. So this is the standard what optimal coding inference. Yeah. Okay, so now we should define something called an inference error. How does the cell know how far it is from its true position, right? And so for that, this is the standard one. Locally, it will compare X star to X, which is the real position, and I'll try to minimize this variance, right? But if I want to do it globally, right, because I want to the whole tissue. So this is the interesting thing. You're doing a control locally, but it's for a tissue level task. Okay, then I want to integrate this over the probability distribution of x's. Okay, so this is the global inference error, and this is a local inference error. And the task here is going to be that you want to minimize this global inference error and make it robust to noise and so on. Yeah, so that's the task. Okay, so is that clear? That was the task at hand. Oh, okay. I missed a slide here which actually showed you the equations for the. Yeah, just 1 minute. How do you go back? Okay. Yeah, trying to go back. Doesn't go back.
The cells that are away from the origin,
Yeah.
will have like minimum information, so
Yeah.
they will produce like maximum noise, right? In the output.
Yeah. So it'll be
like the positional.
right. So the signal to noise ratio will be very large when when you're very far away. So yeah. So then we'll have to see. So of course the cells very far away are going to do worse than the cells. Yeah. I think this is the. Yeah. So very quickly, I just want for some reason I missed this slide. So this is the thing. So in the wing imaginal disk, for instance, it was recorded by these people that the concentration of that ligand here called DPP has this noisy exponential profile. Okay, and the typical equation that people use to solve this is that there's a time derivative of the concentration of the ligand has a diffusion term, a degradation term, and a source term. Diffusion because it's diffusing in the extracellular milieu. Degradation because it binds to surface receptors on the cell surface and internalized. So that's degraded. Okay, and the source, as I said, there is producing cells in the tissue which produce this molecule. Okay, so solution of this typically gives you an exponential profile for large x, and that's the profile that you get, where lambda here is related to the square root of the diffusion coefficient by the degradation rate. Okay, and so if you scale these noisy profiles by that, you find that they roughly fall on the same curve with lambda around 8 cell widths. Okay, so that's the data for the typical data that you get. Right, so that addresses your question now. But despite this, it's going to infer its position to an accuracy of 1 cell width. That's the surprise.
So cell does time averaging.
Sorry.
Cell does time averaging.
Exactly. Cells are also time averaging. We are going to ignore that complication from now. Yeah. Because we're going to take everything steady state. That's related to the question someone else asked about the time.
In the sense that the cell knows about the steady state by just doing time averaging, right? Because it all is just accept this.
So the steady state happens the parameters in this equation, which are measured, are such that it establishes equilibrium very fast. Much faster than the time it's taken for it to read and process the information. Okay. So now we have sort of established our what we want to do. I showed you, I told you what is the objective. It wants to minimize that inference error. I showed you how the profile of the morphogen changes with space. It's a very noisy profile. But now I'll tell you something about what each cell is doing, what the channel is, right? How does the cell process that information? And the cell processes, we're going to take a sort of general model, which will be like a mother of all models, try to encompass all different kinds of scenarios. And we'll say that each cell, which is an information processing unit, can be split up into a reading step, a processing step, a reading and processing and output step. Okay, these are the three things. The reading has to be done by receptors sitting on the cell surface. Typically, we'll allow for them to have two different kinds of receptors. We'll call one as signaling receptors, those receptors that are very specific to the ligand in question, and non-signaling receptors, which could be, which could be any other molecule. I'm just allowing for that possibility, that there are signaling receptors and non-signaling receptors. The number of receptor types are going to declare a branch. Okay, so we'll allow each cell to have many branches, okay? Many receptor types, which will read that information. Once the ligand binds to the receptors on the cell surface, they can be internalized, and they can be internalized into various tiers. We'll call them tiers. This is the first tier. There could be a second tier, third tier, etc., till the output that is generated goes into the nucleus and gives rise to an output theta that I declare. Okay. So you can see that there are many processing steps that we allow for within the cell. We'll call them tiers, and the number of distinct surface receptor types are going to be called branches. Okay. Now, these sets of this kind of machinery you'll provide a chemical reaction network to this. I'll show you some of the chemical reaction networks that we look at, which include feedback. They will also have chemical reaction rates, which I'm going to talk about. And at the end of the day, it's going to give you what the output is for a given input. And then using this machinery, we're going to try to estimate what the inferred position is, okay, through this Bayes theorem. Yeah.
P theta and P theta x.
P theta? This is just the marginal. Yeah, this is just the marginal for that. So the integration for that. P of theta.
Yeah, but how does it will require that to get Px theta?
So this one is what we're going to compute.
Yeah, P theta x is something.
Yeah.
The cell.
Yeah, and once you have that, yeah, once you have that and
something which it knows.
Yeah.
Itself knows.
Yeah.
Px is the prior.
Yeah.
But it also needs to know what is
P of theta. Right. Right.
So how does it know that?
Right. Actually, that turns out not to be too important. But yeah, that's a valid question. You're saying that it's now not a single cell. So it turns out, that this is not very important. Even if I set this to 1, yeah, it's not. Yeah. But we can come to that later. Yeah, that's a.
Madan?
Yeah.
Do these different tiers correspond to different organelles or something inside the cell?
Yeah, exactly. Yeah. So different compartments where this processing happens. And we'll see what is the optimal number of organelles or optimal number of compartments that it needs, etc. Yeah. Okay. So now going here. Oops. So going here, we'll start putting some meat in each of these statements. We'll now start with the surface receptors. What we are going to be doing in this preliminary investigation. Yeah. So what we're going to do in this preliminary investigation is we'll take an open loop control. That is, we'll set the receptors to be outside this optimization problem. So we'll say that the receptors have a profile. This is the psi of x is the signaling receptor, phi of x is the non-signaling receptor, and we'll take it from a class of functions that have either this kind or this kind or this kind or this kind. Okay. So this is my given morphogen gradient, exponentially decaying with noise. Okay. This is the surface receptor profiles, and I choose it from each of these classes. These surface profiles can be parameterized by Hill function. Yeah, and this is technically called an open loop control within control theory. Yeah, so we're not going to put any dynamics to this. This is just provided to us, but we explore all possible functions of phi and psi. Okay, so that's as far as the receptor profiles are concerned. Now we have to get to the channel. Oops, is that the way? Yeah. Okay. So now we have to come to the channel. What happens to the processing units in each cell? And as I said, the processing units are going to be declared via number of branches and number of tiers. Number of branches tells you the number of receptor types on the cell surface, either signaling or non-signaling. Number of tiers tells you how many compartments this processing is going to happen. And so you can write down all kinds of topologies for these multi-branch, multi-tier channels. Okay, here is one receptor type, but everything done at the cell surface. So this is a ligand, the receptor without a ligand binding to this state is a receptor without a ligand. This state is the receptor with a ligand, and this is a degradation. You could have a single receptor type, but two tiers, okay, or three tiers or n tiers. So that is the tier direction, and this is the branch direction. This could be a single receptor type. It could be two different kinds of receptors. One is a signaling receptor and a non-signaling receptor with multiple tiers and so on. Okay, so there are many topologies. So we will now try to look at we'll explore the class of topologies and see how we do. So just to get now each of these, there could also be feedback control. That is, this branch of the network could provide a feedback control to that branch. Okay, so there could be inter-branch feedback controls. Okay, and we're going to allow for this also. So this is the kind of chemical networks we're going to explore. And the idea here is to see which topology gives you the optimal inference error. Okay. Just to have a sort of picture in mind in terms of compartments, the tier 2 branch system has something like this. You have two different receptors, this hairy receptor and this kind of receptor. They bind to this ligand. They internalize. They get internalized within the cell. They meet in some compartment, okay, and then give rise to an output. Yeah. Okay. Now, as I said, each of these chemical networks are associated with a set of equations, which tells you the time variation of the concentration of molecules in that state. Okay. R1 here is the receptor that is bound to the ligand. S1 is the concentration of the non-signaling receptor, which is bound to the ligand. Q1 is the concentration of the conjugate that is having internalized it gets conjugated here. Okay. So this on the right hand side, there are all the sort of chemical processes that are declared by this network. They involve binding to ligand, unbinding to ligand, trafficking or internalization within the cell, conjugation step as I showed you here, and degradation. Okay, these arrows. And so these are simple ODEs, ordinary differential equations, that are maximally bi-molecular. So they are nonlinear ODEs. And then you have to declare what the output is, given that this is the processing step. What is the output? And the output will be given by these purple guys, because the ligand-bound signaling receptors are going to actually provide the output. So basically, you could say R1 plus Q1 is read by the transcription machinery, and that is the output. Okay, and for feedback, as I said, there could be inter-branch, either negative or positive feedbacks. You just renormalize the rate. Okay, for a positive feedback, you increase the rate, which depends on the concentration of molecules there. And for a negative feedback, as in that, you decrease the rate. Okay, and these n's and gammas and alphas and R0, these are all parameters that we're going to optimize over. Yeah, these are called Hill Hill coefficients and okay. Okay, so oops. So that's the process. We fix the morphogen in. Yeah.
The rates did come from phenomenology?
No, so now we're going to. So now we're just doing theory now.
No, I'm saying that this form of R plus R minus.
Yeah, that comes from phenomenology. Yeah, these are the feedback controls. Yeah, so they are typical enzyme kinetics. These are the rates you take. Yeah. There's no formal derivation of that, but there is some sort of approximate. Okay, so just to summarize, this is what the optimization algorithm will be doing. Remember, we want to optimize for the inference error, the global inference error. Okay, so what we do, this is the input. You fix the morphogen input distribution P of L given X. Okay, this is a noisy distribution. You define the channel architecture, those networks that I talked to you about. You define it hierarchically, going from one topology to the other. You first declare the number of tiers, that is number of compartments, and number of branches, that is number of receptor types, and then choose which feedback topology you want to define. Okay? You write down chemical reaction network equations or chemical reaction equations for that given topology. And then that has many, many parameters, namely all the rates that are there in the theory. Okay? So for a given topology, you minimize the inference, the global inference error by over the reaction rates, the receptor concentrations at the cell surface, both the signaling and non-signaling, feedback parameters, etc., etc. Okay, and then you find what is the optimized value of parameters that you get at the end. Yeah? So it's a very high dimensional optimization, right? Because the network can be quite big, and you have many, many, many parameters. But there will be a surprise that that comes in. Okay, so let's go step by step and see what we can do. Let's say that there's only one signaling receptor, and there is very minimal processing. This is the receptor without a ligand. This is the receptor with a ligand. The reaction is going back and forth. There's a degradation term, and let's say there's a feedback on the degradation term. Okay, so I'm going to plot the local inference error versus the position X in the tissue. Okay, and this one here is what you would get when you don't do any processing at all. You're just reading the bare ligand, noisy ligand distribution. This line here, which is slightly reduced compared to this, happens because of this feedback. Okay, so even the slightest amount of feedback does much better than having no no control at all. That is just reading the noisy data. Okay, if you look at what should the receptor profile be in the optimized situation, then you find that this is it. The receptor has to decrease. The single receptor that you have, namely signaling receptor, has to decrease away from the source. Okay. Now let's put in two receptors. Okay. So here is a signaling receptor. Here is a non-signaling receptor, and this is the architecture we're going to look at. Okay. But no feedback. Okay. We're not going to put any feedback here. Again, having two receptors does better than one receptor. It's slightly reduced here compared to the no processing case. However, you see that both signaling receptor and non-signaling receptor decrease away from the source. Okay, they just track the input. Okay. Now that's what we did. Okay. Yeah. So now we'll come to a surprise. Now let's say we have two receptors, a signaling receptor and non-signaling receptor, but we'll include a negative feedback control between this, let's say, between this state and that state. Okay, that immediately brings down the inference error to a huge extent. Okay, in dashed line is the inference error that you would like to have if it was one cell width. If the error was only one cell width, so you want to really bring down that curve to that limit. Okay, but this is pretty good, right? Just having two receptors, signaling and non-signaling receptor, with this negative feedback control, gives you an optimized parameter space for this configuration to have an inference error like that, which is doing pretty well. What's fantastic here is that this makes a prediction. It makes an interesting prediction and a non-trivial prediction of the following sense. This optimized architecture for this topology predicts that the signaling receptor should decrease away from the source. Sorry, the signaling receptor should increase away from the source, whereas the non-signaling receptor should decrease from the source. Okay, compare that to the previous case where both the signaling and non-signaling receptors decreased away from the source. This optimized architecture will predict that the signaling receptor does one thing, namely it goes away. It doesn't track the input, but the non-signaling receptor actually is the one which tracks the input. You can ask what happens if I put multiple receptors, okay? And sorry, you can ask what happens if I put multiple tiers. So if I add one more tier, etc., etc. And what we find is that adding multiple tiers doesn't increase the optimization error by any great amount. So it's too much cost for it. So as long as you have one or two tiers, that's fine. That really does very well. Yeah. Remember, there's two receptors with two tiers. Yeah. That's fine. So, yeah.
There's also an error in the inference error, right?
Yeah.
It's coming from the sample fluxes, fluxes
in the input. Yeah. So this, I forgot to tell you that this is only taking extrinsic noise. This is taking noise in the morphogen signal and not noise in the channel.
So this is given one noisy signal.
Given a noisy signal. Yeah, that's right. That's right. Right? Yeah. Okay. Oops.
Like for example, like if you see that like 2 or 3 points are way off, right?
Yeah.
And then how is it possible that cell actually decide that is the 7th cell has to have something and the 9th cell has to have something? I'm not saying that given that so much error fluctuations.
That's right. That's right. So clearly there is also some temporal averaging going on. There could be some spatial averaging. We'll come to all these. So the story I'm going to tell you today is a preliminary story. It opens up lots of questions, but it's a new way of looking at this very important question. Yeah. So far, as I said, we only included the extrinsic noise. What happens when you include intrinsic noise, that is noise within the channel? And this is where the number of tiers comes in. It turns out that adding extra tiers actually buffers the nonlinear effects of this feedback. So you find that by adding in some sense, what we can show is that by adding extra tiers, further tiers, away from this thing can buffer the noise in earlier tiers. Okay? It does so by actually, for instance, if I look, we have a nice dynamical theory argument, which a dynamical systems argument, which shows that if I look at the variance of the concentrations in these two signaling states, so the variance of this plus this, that of course is equal to the variance of the individual persons plus this covariance term. But this feedback gives you a negative value for this covariance so that it reduces the net variance of this. Okay, and that's the interesting thing. That's what feedback does here. So by adding more tiers with feedback, you actually buffer the noise in each. Okay, so now how does this do in terms of experiments? Oops. Right, that's the.
If you had a positive feedback, would the covariance be positive and would that increase the error?
That's right. So right. So now, Chaitra in Jitu's lab had been working on trying to look at the profiles of all kinds of molecules in a tissue at different sections. Okay, so this usually what people had done was to look at concentrations of the ligand in here, okay, and concentrations of the receptor in the top slice of this epithelial sheet. But what Chaitra did was to do it in several sections, in 8 different sections, okay, and therefore was able to compute the concentrations of the ligand on the surface, in the initial tiers, in different tiers, right? Very, a fantastic task that she had accomplished. And in collaboration with her, we have been able to sort of verify some of the predictions that we make here, especially the prediction having to do with the opposing profiles of the signaling and non-signaling receptor. Okay, so what Chaitra has been looking at is the wing imaginal disk, and she's looking at a ligand called wingless. Okay, and wingless, as I said, has this kind of profile. Okay, this is the profile of wingless at the cell surface here. You can see that the noise is pretty large, even at these tails. This is a question that you're asking. Yeah, and this is what wingless does at each cell. Wingless has two kinds of receptors on the cell surface. One is a signaling receptor called frizzled. The other is a non-signaling receptor called DALI. Once DALI and frizzled bind to the ligand, they get internalized. Okay, and then they meet in the common endosome, whereupon signaling for transcription happens. Okay, so this you can see is very similar to our two branch architecture. Okay, and so we can apply the results of the two branch architecture to the system. Yeah. Okay, so that's the two branch architecture that I'm talking about. Okay, one branch is here, the other branch is here. Okay, like so. This is the signaling receptor, this is the non-signaling receptor, and they meet in the common endosome. Okay, so this seems to be a prime example of our two branch system. And remember what the prediction of this optimized two branch system was. It said that the signaling receptor in blue, okay, increased away from the source, whereas a non-signaling receptor, in this particular case is DALI, decreases away from the source. And this is precisely what you see in the Drosophila wing imaginal disc. Chaitra plots here this is the surface profiles of frizzled, the signaling receptor. This is the surface profile of DALI. You can see that it has qualitatively the same kind of feature as the prediction here. Okay, she then looked at the coefficient of variation, which is the ratio of the variance to the mean, in each cell as a function of position. Okay, and this is what you get. This here is what we get from our theory. Okay, for the optimized channel, and here is what Chaitra sees in her experiments. Okay, now one of the predictions in the theory is the following: if we get rid of this branch here, namely the non-signaling receptor, okay, then you should get a very bad estimate of the inference, or you should get a very high CV. Okay, and that's what we see. This is just to say that this is what we see in theory, and this would be a prediction that we make for the experiments. Chaitra then could experimentally remove this branch by using a dominant negative of a major player in this pathway that cuts off this branch, only leaving the signaling pathway. And indeed, what she sees is that the coefficient of variation increases like so. Okay, so that seems to be a nice prediction that has some level of qualitative variation. We are now trying to do these things quantitatively by taking her data and then going through the kind of process that we have done so far. Now, this makes. I just want to end with the following set of observations. This has to do with the geometry of the inference landscape. Think of the inference error as being like a free energy, and just like you would plot a free energy landscape, I could plot an inference error landscape. Okay, and what happens is that I typically, by this optimization, I've gone to the minima of that inference error, just like you'd go to the minima of a free energy. But then I can ask how, what is the curvature at that minima? Right? Are there flat regions? Are there flat directions, or are they very highly curved directions? If I go to the optimized parameters and change the parameters ever so slightly, does the inference error increase hugely, or doesn't it change too much? Okay, so this is this analysis of the geometry of inference landscape gives rise to something called stiff and sloppy directions. okay, or stiff and sloppy parameters. So you're moving in parameter space, and you declare those directions as being stiff if you change it slightly, the inference error changes by a huge amount. So the cell wants to keep that under control. Okay, so those directions are stiff. Sloppy directions are ones that if I vary them, then it doesn't matter really, right? So this is very much like relevant and irrelevant variables in reinforcement group theory. Okay, and so this is what we would like to try to investigate. And it turns out from a lots of different kinds of analysis, what we find is that the concentrations of the signaling receptor and their binding unbinding properties are very tightly controlled. Okay, if I go to the optimum of that and change things slightly, the inference error increases tremendously. So this is a prediction that again that one can verify that the signaling receptor is tightly controlled, it's very stiff. However, the non-signaling receptor is allowed to be sloppy. Okay, if I vary, so for instance, this is the deviation of the non-signaling receptor from the optimum, and this is the deviation of the signaling receptor from the optimum. This is a local analysis of the geometry of the landscape. And you can see that in the direction of the signaling receptor, okay, you change the inference error. But in the direction of the non-signaling receptor, you make hardly any change in the inference error. Okay, this is replicated in the binding rates. The binding rates of the non-signaling receptor are pretty broad. I can allow for variation in the binding rates to be anything. The inference error doesn't change too much. But the binding rate in the signaling receptor is tightly controlled. It hugs the sort of right edge of the allowed range. Okay. And this is interesting because it's known that the non-signaling receptor of wingless, namely DALI, is very promiscuous. It can bind to many, many different ligands. Okay? And that's consistent with this broad distribution of binding rates. Yeah. Okay. This is roughly the same thing that I want to say. This is what was previously done was a local analysis. This is a global analysis. After all, this is a non-convex optimization, so there are many, many minima. And I can say where do the minima of this inference error lie? Do they lie in the directions in the sloppy directions, or do they lie in the stiff directions? And what we find here is that indeed the topography of this inference landscape is like this Himalayan range. Okay, many sloppy directions. Okay, and all the minima of this, all the various discrete minima of this inference error lie in this direction, and there are very, very few stiff directions. Yeah, so this turns out to be an interesting feature of such a high-dimensional optimization.
Do you understand why?
This is what we'd like to do. I can give you a hand-waving argument because we've seen this in many different kinds of optimizations. And this, as you're saying, it requires us to explain this. And so one possibility is that this dimensional reduction that happens, I mean, this is not only us, but many people have seen, and this is famously called a dimensional reduction problem. And so what the cell wants to do is to have, by having, I'll give a plausible argument. By having a few number of stiff directions, the cell needs to have only a few parameters to control. Each parameter, each control comes with a cost, and so it doesn't want to expend too much cost in controlling everything. That is one. But the second is that by having these sloppy directions, it also allows for evolvability. And so, but these are stories, post-facto stories, and so yeah. But it would be interesting to try to see. I think the question you're asking is, what is the generic feature of such a system which allow for this kind of dimensional reduction? And we don't know that yet. Yeah. Right, right. Right. Exactly. I don't know if we can throw away, but but it doesn't matter over a time. Yeah. Yeah. Exactly. Yeah, exactly.
But I thought you showed that these sloppy directions are required. In absence of that, the error.
Yeah, that's what I'm saying. I don't. Yeah.
It
Yeah, I'm not saying that you can't throw away, exactly. So clearly you need the, all I'm saying is it's flat. But you of course, you need the stop redirections. You need the non-signaling receptor. Yeah. Yeah, okay. So what are the future directions? The major future direction that we think is to bring back the receptors into a closed loop. That is, have a dynamics of the surface receptors, which will then couple to the ligand, noisy ligand outside, and therefore have the possibility of sculpted back. It's sort of interesting that you remember what I said about the dynamics of the ligand. The dynamics of the ligand has a diffusion term and has a degradation term. The degradation term comes from the fact that the ligand can bind to a receptor. So but the receptor is also required for reading, right? So the receptor is doing two things: is carving out, is sculpting the ligand, but is also reading the ligand. Okay, so these two, so clearly then there's an interesting feedback that should be explored, and this is what we are doing now. In some preliminary work that Krishnan has done, he does find that by putting a negative feedback between the output and the receptor concentration and the non-signaling receptor concentration and the positive feedback for the other one, for the signaling receptor, you do recover these opposing profiles of the receptor. So we are moving in that direction to try to see if we can get a dynamical theory for this. This one I just talked about, the emergence of a small number of stiff directions. This seems to us to be a very important general problem that we need to investigate. And of course, that the position inference is also done when in the context where the tissue is growing. And so when you have a dynamically, a tissue which is increasing in size, how does the cells find infer their position? So that's another interesting question. Okay. Finally, I'll just take 1 minute. Is that okay? That's right. Yeah. Finally, I just wanted to state one thing here, that this decoding molecular information problem is sort of is an interesting way to look at this information processing thing, where this is the following thing. You have a morphogen source, which I'm just calling X, yeah, and then you have a channel which is reading this noisy morphogen signal, the signal coming from X, and then it gives rise to an output or the inference of the position, which is the receiver. The questions that we're asking is that given this, given the noisy morphogen source, what is the characteristics of the channel that gives rise to some global optimization? Okay, so it's flipping Shannon's question on in a different way, where in Shannon you're given the nature of the channel is given to you, right? Here we are trying to determine what is the optimized channel that will give rise to this kind of optimization, no? Yeah, global optimization. Mm. Yeah. So, right. Okay. So then that's just to end the other another paper which sort of talks about encoding of molecular information has the same philosophical basis. I mean, if any of you are interested, they can discuss this, or you could read the paper. These are the people who have done all the work. A brilliant student, Krishnan, who is now who will be going to IST in Austria to do his postdoc, and Chaitra, a brilliant student of Jitu's, who is now at Harvard doing a postdoc. And that, of course, is Jitu, many of you know. Before I end, I just wanted to put this and we can take questions. We just started in collaboration with ICTS. NCBS and ICTS have a formal program called the Physics of Life, where we just started a whole graduate program on this, and we are inviting students, physics students, to physics, maths, and computer science students to come and go be part of the program. And there's also an advertisement for postdocs at NCBS. We now have 7 theorists in the Simon Center at NCBS, and it's a very exciting place to be. So that's it.
Any questions?
So, when Shannon derived his proof, he assumed the maximum entropy state for the strings that were passing through the noisy channel.
Right.
But in your case, is it fair to assume that? I'm not sure. I'm unable to think fast. I don't know enough. So maybe you can help.
Right. I'm not sure I can
probably.
Think fast.
Do you see a role for compressed sensing coming in here? I don't. Since I don't expect a maximum entropy states coming up here, and there is sparsity in the information that is coming because the individual morphogens that are coming, the signaling is sparse and it's also coherent. The two conditions for compressed sensing are being met. Do you think that would actually improve your fidelity?
I haven't thought about it, but we have talked off and on about compressed sensing and its appearance in such situation, but we should talk about this. Yeah.
So wondering, is there a way for one cell to know, what a neighbor, how much morphogen a neighboring cell?
Right. So remember, so this precise question will be addressed in our dynamical theory, where we allow the surface receptor profiles to be dynamic and read the information on the outside. So you could imagine several scenarios. You could imagine that the surface receptor profiles in neighboring cells could be correlated with each other as a result of mechanical interaction or even signaling. Okay, that's one. You could also imagine that if the surface receptors in this cell are allowed to talk or couple, or they sense the ligand concentration and sculpt them, because there's a diffusion term in the ligand concentration, that will lead to a natural coupling between these two. So in fact, that's what we see in our first attempt to try to address this question. But it's a very interesting question because there could be other possibilities. Because it turns out that it could be that after all, the surface profile of a receptor is set by the rate of internalization into the cell. Right? If you have very fast internalization, then there'll be lower surface profile. It could be that the rate of internalization is called endocytic rate. Could be in neighboring cells could be coupled because there's a tension that this applies, and endocytic rates are very strongly sensitive to tension. So there could be a role for mechanics.
But experimentally, nothing is known.
In this context, it's not known, but people know that the endocytic rates are sensitive to tension for a single cell.
Oh, okay.
These kinds of experiments have not been done in tissues where you have collections.
Thanks.
Questions?
This analysis, like this P of X, you anyway take it to be 1. And you said that P theta is not very important, right? So it's basically Px given theta is same as P theta given X, right? So therefore, I mean, are you saying that given theta, it essentially knows where the cell is, right? In this assumption.
Exactly.
So it's some function of theta minus X or something like that, right?
Exactly.
Okay. And
yeah.
So where is this information stored in the cell that given theta is like, what is the equivalent of Mathematica within the cell? Basically, right?
So clearly, of course, we are silent about it, right? Yeah. But that's an important question to ask. And it means sort of trying to get into the nitty-gritty of what is really happening. So the simplest answer is I don't know, but one could imagine that that let's say when it comes to the sorting endosome, then there's a signaling that happens, which means some molecule is produced, And that molecule, in many cases it happens that these molecules then shuttle to the nucleus. Okay, they go to the nucleus. So then once it's there in the nucleus, it could either activate a transcription factor, which then turns on a gene, and the gene then makes those hairs or whatever, makes a certain feature of that cell. So that is, you've now declared what its position is, because that's where it has to be. You see?
Very simplistic picture. Does it mean that given a theta, basically whether there will be a hair or not in that cell? Okay, you are getting this thing and it's producing theta, and given theta within some threshold, it will produce something, right?
Yeah.
Is it very simplistic picture, or it's
yeah? I mean, it's as simplistic as what we have done also. We just.
No, but then I don't have to do any theory, right? In the sense that, okay, given this theta, I have a hair, or given this theta, I have a finger, or something like that, right?
Yeah, but I don't know the architectures. I don't know what the architecture is inside. The goal of this was to say what is the optimized architecture. That is, the goal of this was to say that if you look at a system which has two tiers, or let's say, if you look at something like the wingless this thing, which has a this thing architecture, then this is the architecture, optimized architecture, and this is how it looks like. So here we're just saying that we're talking about the existence of this optimized architecture. That's what we found out in this. And the next step is to try to see whether this optimized architecture can be arrived at through a dynamical theory. I think what you're asking is that given, if I know what the optimized architecture is, then it's a simple question of computing theta, and then but that's what we're trying to say. I mean, a priori, I don't know what the architecture is. So I'm starting off my optimization with exploring all possible architectures, and I'm finding that in this particular case, it optimizes the inference error. In each problem, I can compute theta if I know all the intermediate steps. Just a set of ODEs that it has to do. And then I want to ask whether that inference is robust to noise. So noise both in the outside and in the internal machinery.
Okay, let's thank Professor Rao for the nice talk.